Hello, my online learners. Part 3, digestion. Let's continue on our journey. When I left you last, we had looked at the organs of the digestive system, starting at the mouth, how the food gets taken from the mouth to the pharynx, down the esophagus, into the stomach. And we talked about the small intestine in terms of that's a place where digestion and absorption occurs. And then we talked about how the material moves into the large intestine, and that's where water is reabsorbed back into the bloodstream, and also where there's the gut biome that adds the bacteria that adds nutrients and vitamins, including vitamin K, to your body. But we didn't mention the accessory organs in terms of the liver that creates bile, and it's stored in the pancreas. Let's try that again. The liver, which creates the bile, it's stored in the gallbladder, and then the pancreas, which makes all sorts of gastric juices, and we'll break down all the gastric juices. But first off, let's look at the liver. When we look at the liver is very large, and it takes up the majority of the top of the abdominal cavity, and it has two lobes, the left and the right lobe, and it's divided by this falciform ligament. And underneath the liver, usually tucked quite tight underneath the liver, is the gallbladder. And the gallbladder's main job is to store the bile. Now, the liver does a great many things, and we're only going to look at the digestive property of the liver, and that is that it creates bile. When you look at the liver itself, the liver has a very good blood supply going to it. We talked initially when we talked at the circulatory system how the liver has this hepatic portal vein which takes all the blood off of the guts, the mesentery, or the large and small intestine, and it passes it through the liver for cleaning and detoxification. That is one function of the liver. The other function we're going to look at is how the liver itself produces something called bile salts. So when you look at the liver, the cell tissue of the liver is beautiful. It's these nice hexagonal shapes with lots of portals in it. It's got these sinusoids, so it's very, very spongy. If you ever get the opportunity to look at real liver, fresh liver, you may have cooked it. It's a delicacy. I used to eat a lot of liver when I was a kid, and then I went to university and I studied the liver in large amounts of detail, and now I have difficulty eating it without seeing the little sodium channels and the hepatocytes, and what education will do to my mind. Anyway, when you look at it, there's a central canal where the blood will go through and it helps filter it, and then one of the process is to make bile salts, and then the bile salts are shown in green here, and then the bile salts are collected, and if you look, so as the blood flows through the liver, you've got the portal veins carry the rich nutrients blood. The hepatic artery branches to carry oxygen-rich blood. The sinusoids filter the blood. There's a type of macrophage in there, a specialized one called a Kupffer cell, which removes the blood and removes the debris and bacteria. Then the central vein carries the processed blood, and the canicula are the ones that carry the bile to the hepatic ducts. So the central vein kind of goes the opposite way of the canicula. So when we look at the top here, this is where the hepatic ducts come. So the bile that is created in the liver comes down, and if it can't go directly into the duodenum, so here we have the small intestine, the part right off of the stomach called the duodenum. And if the bile cannot enter at this time, it gets stored in the gallbladder, remembering that the gallbladder is tucked under the liver. So here's a good example to show how the gallbladder is tucked under the liver. So the bile will be stored in the gallbladder. When there is food in the duodenum, it will trigger an enzyme called cholecystokinin, which I'll type up later for you, to cause, an enzyme, sorry, a hormone, cholecystokinin, which will go in the blood supply. That's why it's a hormone. And that will cause the gallbladder to contract. And when it contracts, it will then push the bile salts, kind of interchangeable words, through the cystic duct into the common bile duct. And the common bile duct will then dump into the small intestine or the duodenum. Now, at the base of the common bile duct is where there is the junction of the pancreas. Remember when we're talking about hormones of the pancreas and the whole endocrine, there's an endocrine function or a hormone function where the hormones produced in the pancreas, the insulin and the glucagon, will go into the blood. That is the endocrine process. The exocrine process is when you have your pancreatic juices. Now, your pancreatic juices have different stuff, which I'll show you the textbook so you have all the detail. But when you look at the body of the pancreas, there's all these little acinar cells, and the acinar cells will then secrete. It goes into the duct, and then the duct will join with the common bile duct, and it will be dumped into the duodenum. Okay, so when we look at the duodenum, this is the site of most of the digestive processes, because all those juices coming from the pancreas add for further digestion. Then when you look at it, there's also the jejunum, which is the site of absorption, and then you've got the ileum, which has the Peyer's patch, which, if you remember back, are lymphatic tissue to help with bacteria. Okay, so now let's look at, I want to show you a bit of the video again, so this way you can look at the structure of the small intestine, and we'll also look a little bit about how the juices dump in, and then we'll summarize the juices.
These increase the surface area of the gut, making it easier to absorb nutrients. First, the pancreas pumps out a juice that neutralizes stomach acid. Then bile from the liver breaks down the fats into tiny droplets. Smaller droplets are easier for the intestine to absorb. After an hour and a half, the small intestine has absorbed most of the nutrients from the food. It's time for what remains to move on.
Okay, let's look at what actually we mean when we say, okay, the introduction in the textbook says, although the liver, gallbladder, and pancreas are accessory organs of the digestive system, they play a crucial role in the digestive process. Each of these organs secrete digestive fluid or enzymes into the digestive tract at the junction of the stomach and the small intestine. The liver is the largest gland. The liver fills up the upper right abdomen below the diaphragm. Even more important than the size is its functions. The liver performs over 250 tasks, including storing and releasing glucose, processing vitamins and minerals, filtering toxins, and recycling old blood cells. In this chapter, we're going to focus on the liver's role in digestion. And the liver has 4 lobes, 2 on each side. That's not really important, but there we go. And then we talked about underneath and how we have the hepatic portal, which is filtering the blood from the intestines. How there's sinusoids. How there's a blending of the blood. And how the liver is creating bile. And the bile travels in the canalicula. Okay, oops. Good. Now, the liver can go down and it will store into the bile duct. Good. So, let's add that to our enzymes before we get on to the pancreas, because the pancreas is very confusing. So, here we are. with our table. It's got to be a way for me to center this just a second. There we go. So we talked about that. Let's now talk about the liver. The liver produces bile. Now, the thing about bile is it is not a chemical. It is what's called an emulsifying agent. So it will emulsify, which is a way of making big droplets into little droplets. It emulsifies fat, and I can't spell today. It emulsifies fat into smaller droplets. Now, because it's making it smaller droplets, that means there'll be more surface area. So this way, the other enzymes that are coming from the pancreas can act on it. Good. We've got our liver taken care of. Let's put our pancreas in here. Now, notice there's no E at the end of pancreas. And there's a bunch of them. So the first thing we're going to look at is a neutralizer called bicarbonate ions, or sodium bicarbonate. And this is to neutralize the acid chyme. And remember that chyme is the fancy word for contents coming out of the stomach. And it will neutralize it, which will shut off some of the stomach enzymes, and then will turn on the other enzymes. Okay, back to the textbook. So back here in this one, epithelial cells in the pancreatic duct secrete sodium bicarbonate, which buffers the highly acidic chyme entering the duodenum from the stomach. So that's number one. Then we talk about the acinar cells secrete enzymes in an inactive form. Again, it's important they're inactive so that they don't digest the tissues that make them. And enzymes that help break down lipids, proteins, and carbohydrates. So there's many enzymes. Let's look at those enzymes. Okay. We talked about carbohydrates, and we talked about how carbohydrates, the one main carbohydrate we're breaking down is starch. Now, we've already talked about how there is amylase in the saliva, which will hydrolyze, which is the fancy word for breakdown using water. And then the salivary amylase becomes inactive in the stomach because of the low pH. Then, when we get to the pancreas, when the chyme reaches the small intestine, it mixes with pancreatic amylase, and the process of starch digestion resumes. So we've got pancreatic amylase. Let's finish this one plate, and then we'll go on and summarize. The final steps of carbohydrate digestion take place at the level of the villi. The membranes of the epithelial cells covering the villi contain the enzymes sucrase, lactase, and maltase. Notice they both end with ASC ending, so that it just means it's an enzyme for what the beginning of. So sucrase breaks down sucrose, which is a disaccharide. Lactase breaks down lactose. Maltase breaks down maltose. So there's enzymes that go into the final process to break down carbohydrates. Okay. Summarize. So we have our bicarbonate, then we have our pancreatic amylase. So it is created in the pancreas, acts in the duodenum. Okay, and since it's an amylase, it continues the breakdown of starch to simple sugars. Okay. Now, I'm not going to include the sucrase and amylase because they aren't that tricky because basically you have the word ase at the end of them. So that is the breakdown of our carbohydrates. Let's continue on. In the next breakdown, we're going to look at our proteins. So we talked about pepsinogen being released by the stomach cells, the chief cells. Proteins are complicated molecules consisted of folding and twisted amino acids linked together by peptide bonds. Before the body can absorb the amino acids, it must break the peptide bond, and it does it with enzymes called proteases. Notice the ASE ending, meaning enzymes, on the protes or the proteins. So you have pepsin, which we talked about when we talked about the stomach, and the pepsin, well, it's actually pepsinogen, and the pepsinogen will be turned into pepsin in the stomach. Now, there is a pancreatic enzyme that's actually called trypsinogen, and the trypsinogen will act and can be converted into trypsin once it gets into the duodenum, and chymotrypsin, which once it gets into the duodenum. So, and they break down proteins, and then you have something called peptidases, but it ends in ase, so that means it's going to break down peptides. So that's not as tricky. So let's look at our pancreas. So it's created in the pancreas, acts in duodenum. So we have our, what did I say the words were? Trypsin. And I'm going to put trypsinogen, just so you know it starts in the inactive form and then it becomes trypsin once it gets into the duodenum. Now, because the duodenum is now more neutral, the neutralization of the chyme will activate it from trypsinogen to trypsin. And that will continue the breakdown of proteins. Okay. And then we have chymotrypsinogen, which becomes chymotrypsin. And that is released from the pancreas. And it acts in duodenum. And that will continue breaking down proteins. Okay, now I'm not going to use the word proteases because proteases continue to break down proteins and peptidases break down peptides. We need a few more inserts below. We need a few more rows here. Good. Hit the save button in case you crash. Excellent. So we've looked at the breaking down of proteins using pepsin and trypsin. Now, when we look at fats, there's a couple of steps. We looked at bile being released by the liver, stored in the gallbladder, and it will act on droplets of fats, breaking it down to droplets. We've already included that in the process called emulsification. Next box, once the fat is emulsified, pancreatic lipase, and here's one where you have ASE, so it's an enzyme that breaks down lipase, begins to digest the fat. In the process, fats are broken down into a mixture of glycerol, short-chain fatty acids, and monoglycerides. Then the monoglycerides are absorbed they're too large to be absorbed in the capillaries, which they go into the lacteal of the villi. Very important there. Okay, so let's summarize. So it's pancreatic lipase, which is produced in the pancreas. And then it acts in the duodenum. And it breaks down lipids. Okay, good. Now realize here that the lipids will, let's put down into glycerol and fatty acids just so you know what's going on. The long chain fatty acids go in the lacteal, but let's put it down so we know. Breaks down lipids, and let's just use an arrow because why use words if you don't have to. And it breaks it into glycerol and fatty acids. Okay, and the fatty acids that can't go into the blood supply will go into the lacteal. Good. That's lovely summarizing. Lovely summarization. Good job. Okay, so now we've broken down all our bits, and all the small bits go into the small intestine go into the capillaries of the small intestine, with the exception of the long chain fats, which go into the villi. The lacteal of the villi. Good. That's the small intestine. Digestion occurs in the small intestine. Okay, let's look at the large intestine. Once the food is processed, once the food has been processed and its nutrients absorbed, the remaining residue is ready to leave the small intestine for the large intestine. In fact, 500 milliliters of residue consisting of undigested food, sloughed off epithelial cells, mineral salts, and bacteria enter the large intestine every day. So that's what, 2 cups or a medium-sized yogurt container. The large intestine absorbs large amounts of water from the residue by passing the resulting material waste or feces out the body. Okay? Following figure shows different regions of the colon. Food enters one end, leaves the other. That's pretty much all we need to know there. But notice, one thing too that's on here is this is where your appendix is. Your appendix is down. So you have the small intestine, which is the ileum, joining the cecum, which is kind of this blind-ended pocket. So this is the ileocecal valve, which is a sphincter muscle. And then you have the appendix. There's a large amount of lymphoid tissue here. Then you have the ascending, transverse, descending, and the rectum. You also have the anal canal, which is the last inch, and this is the opening. It contains an internal sphincter, which is involuntary, and an external sphincter, which is voluntary, and it is skeletal muscle. So the interior involuntary smooth muscle, the exterior voluntary skeletal muscle. Okay. Good. Now again, here's a nice summary of your enzymes and what's breaking down what. And that is the end of chapter 21, which is the digestion. Pay attention to where lipids are broken down and where they're absorbed. Pay attention to where carbohydrates are broken down and where they absorb. Pay attention to where proteins are broken down and where they are absorbed. Okay? And so basically know your parts, know what they do, and you may want to make a summary table of part and function. So part, description, and function, and talk about it. And that's the end of chapter. That's the end of part 3. We have a part 4 talking about chapter 2. So stay tuned or review this stuff because that was a lot of work.